Is Dr. Stephen Quay. He's a physician and a scientist with hundreds of published scientific articles. Dr. Quay has been cited over 10,000 times, holds 87 patents across 22 different fields of medicine. He's invented 7 different FDA-approved medicines. Further, he conducted an early analysis of COVID-19, which has been downloaded nearly 200,000 times. He's an expert in the field and brings an upper echelon of analysis to the question we're posing today about the origins. Now, to you, Dr. Quay.
Thank you, Congressman Scalise and Comer, members of the subcommittee of the coronavirus crisis, ladies and gentlemen. I appreciate the nonpartisan approach the subcommittee is taking today. Clearly, science in the last few years, especially on the topics related to the COVID pandemic, has been co-opted by geopolitics. Thus, I am here not as a mouthpiece for any particular party, but as an American scientist. I dedicate my testimony today to the more than 600,000 Americans that this pandemic has killed so far, with the hope that by clarifying the origin of COVID, we can help to prevent future pandemics and the loss of innocent lives. Given this backdrop, I will be keeping my remarks today to matters of science. I'll state facts and evidence about the pandemic that I have collected. I'll explain why those facts lead to certain conclusions, allow a particular inference, rule out a hypothesis about the origin. Today's testimony is an effort to answer the question posed on December 30th, 2019 by Dr. Tukuafu-Sheehan, head of coronavirus research at the Wuhan Institute of Virology. She had just been told that a novel coronavirus had caused an outbreak of pneumonia at hospitals close to her laboratory in Wuhan. In what I believe was her last unscripted public utterance, she has been quoted as saying, "Could they have come from our lab?" In 18 months, we've learned an intense amount about the origin of the pandemic. But one of my frustrations is that virologists and science writers around the world seem to want to ignore what has been learned and the inevitable consequence it reveals. As inconvenient as it is, I believe the evidence conclusively establishes that the COVID pandemic was not a natural process, but instead came from a laboratory in Wuhan, China, and it has the fingerprints of genetic manipulation for a process called gain-of-function research. Undisputed facts that support this hypothesis. Now, as a reminder, the top left there, a little science. As a reminder, all zoonoses are diseases that involve a virus infecting an animal, the animal coming in contact with a human, and then the human being infected. The question of the origin of a zoonosis is the question of the location of the animal. In natural zoonoses, the animal is in nature, a cave, a farm, or the market. The infected human comes in contact with the animal and gets sick. For lab-acquired zoonoses, the animal is in a laboratory or in cells from an animal in a petri dish, and the human works in that lab. In deciding between a natural origin and the lab origin, there are 6 facts no one disagrees with. Number 1, the Chinese first told the world that the COVID pandemic began in the Hunan seafood market because approximately 1/2 of the early cases had been associated with that location. This would have been reminiscent of the 2 previous corona epidemics, as both SARS-1 and MERS began in live animal markets. However, after 18 months of investigation, we now know it did not begin in a market in Wuhan for the following 3 reasons. First, none of the 11 patients from the Hunan market or any other market in Wuhan were infected with the earliest virus, meaning they came into the market already infected. The 4 patients with the earliest genetic version of the virus all had one thing in common. None had any exposure to the Hunan market. Second, we know in January 2020 from public scientific databases that the patient with the earliest first complete virus sequence was a 39-year-old man treated at the General Hospital of Central Theater Command of the People's Liberation Army of China, located on Wuhou Road in the Wuchang district of Wuhan, about 3 kilometers from the Wuhan Institute of Virology. After 18 months and almost 3 million viruses sequenced worldwide, this virus still remains the earliest. Second, none of the environmental specimens from the market had the earliest virus, meaning they also came into the market. This included 1,055 samples of frozen food in the market, all of which tested negative for COVID. Third, 457 animals from Hunan market were tested. All were negative for COVID. 616 animals from suppliers to the market were tested. All were negative. 1,864 wild animals from southern China of the type found in the market were tested. They were also all negative. These data from the publicly available WHO report established that it did not begin in the Hunan market or any other market in Wuhan. Number 2, failing to find it in any market in Wuhan, the search was expanded to all of China after testing 80,000 samples from 209 different species from other markets, farms, and nature. The virus has not been found in a single specimen. The probability of this for a community-acquired infection is about 1 in a million. This is what you'd expect for a lab-acquired infection. Number 3, after testing 9,952 stored human blood specimens from hospitals in Wuhan from before December 29th, there was not a single case of COVID in any specimen. It was expected that between 100 and 400 would be positive. The probability of this for a community human-acquired infection is also about 1 in a million. But this is what you'd expect for a lab-acquired infection. There is no evidence of multiple animal-to-human transmissions. With 2 prior coronavirus outbreaks, SARS-1 and MERS, 50 to 90% of early cases were from various animal-to-human infections. For SARS-CoV-2, 249 early cases of COVID-19 were examined genetically, and they all were human-to-human transmission. For a community-acquired infection, this is the probability of tossing a coin 249 times and getting heads every single time. This is, however, what you'd expect for a lab-acquired infection. Number 5, SARS-CoV-2 has a unique trigger on the surface called a furin cleavage site and a unique code in the genes for that site called a CGG dimer. So these are 2 independent levels of uniqueness. The furin site is why the virus is so transmissible, why it invades the heart, the brain, and the blood vessels. Viruses like Ebola, HIV, Zika, yellow fever all use furin sites, and this is part of the reason for their deadliness. But the entire group of coronaviruses in which SARS-CoV-2 belongs does not contain a single example of a furin site or a single example of the CGG dimer code. A comprehensive study by an MIT group shows that there has never been a furin site or the CGG letter dimer in this group of coronaviruses for at least 1,000 years, since William won the Battle of Hastings in 1066 to become monarch of England. But since 1992, in gain-of-function experiments, the WIV and other laboratories around the world have inserted furin sites into viruses repeatedly. This is the only sure method that always works and always makes them more infectious. And the CGG code found in COVID-19 is commonly used in laboratories around the world, including the WIV. You can literally order it from a supply company on the internet.
That's bad. That's way bad.